Now we're going to talk about the last step, which is called translation. In both prokaryotes, that's what this cell is. You see there's no nucleus. Prokaryotic cells and eukaryotic cells. This one on the right is eukaryotic with the nucleus. Translation happens in the cytoplasm, and that's because that's where the ribosomes reside. So the ribosomes are the machinery that does the translation. You know, when we learned about cells and we said ribosomes build polypeptides, or you could say ribosomes build proteins. That's this process of translation. And you can see it's very similar in both of bacteria and eukaryotes, at least how we're talking about it, because we're keeping it really simple. There are three main stages. They actually have the same names as the transcription stages: initiation, elongation, and termination. Basically, initiation is like getting the ribosome on the mRNA. Elongation is making the long polypeptide, and termination is stopping making the polypeptide and having everything disassemble. Here's an overview of the whole process here. You can see the ribosome, the tRNA with an anticodon and an amino acid attached. You can see the codons and on the mRNA, and you can see how this tRNA is like an in-between. It's the connector of the amino acid and the mRNA codon. So that's how those get matched up, is through this tRNA intermediate. And then we have this long polypeptide growing out of the ribosome, and that's what you see here, this little polypeptide. And eventually that will be released, folded into a protein. tRNA stands for transfer RNA. Each amino acid that's loaded on is specific to that tRNA. So anytime you have a tRNA with a certain anticodon, we'll see that in a moment, a certain anticodon matches with a specific amino acid. And like I said, that's the connector between the mRNA and the amino acids is the tRNA. tRNA has a three-dimensional shape. You can see this in the middle picture, which allows it to do its job. And in the book, you see on the right side here, this is like our representation, a symbol used in the book. It has the most important parts. So one end, which is the same as this part or this part on these two more molecular structures, that's where the amino acid is going to attach. On the other end, you can see there's three on this little loop at the bottom. There's these three nucleotides. These are RNA nucleotides. That's called the anticodon. And anti means opposite, so it's the opposite of the codon. It's complementary. And remember, the codon is that sequence of mRNA. You can see in the folded structure, the anticodon sticks out in this loop, which is important because that's how it touches the mRNA. And it's, you see the rest of these are not, the rest of the nucleotides are not near that. So it's just those three that match up with the mRNA. Importantly, we have polarity on the tRNA. So a 5' end and a 3' end. Just like all, or I should say almost all RNA is single-stranded. It's all single-stranded when it's made, at least. And this one stays single-stranded, but you can see it folds into this, like I mentioned, three-dimensional structure through hydrogen bonds. There's partial negatives and partial positives on these nucleotides of the tRNA, and they allow these guys to attach and to form this structure that's really important. In the initiation part of translation, there are sort of two steps. One, you're going to match the tRNA and the amino acid. So you have that specific anticodon with a specific amino acid. Of course, there's an enzyme because this is a reaction. Its name is amino acyltRNA synthetase. That's the general name. So basically, you can think of it as the amino acid tRNA synthesizing or attaching ACE. Remember, that's enzyme. So it's loading that correct amino acid onto the correct tRNA. You can see here, this is the amino acyltRNA synthetase, but it's a tyrosyl because it specifically attaches the amino acid tyrosine, which they're representing in this hexagon, to the correct tRNA, which has the AUA anticodon. So only that tyrosine tRNA can fit in this part of the active site, and only the tyrosine amino acid can fit in this part of the active site. And so then through a reaction using energy, it's going to attach that amino acid to that tRNA, and now you have the particular match. And it does this there's different enzymes for the different amino acids and different tRNAs, and so each one gets matched up because the active site's different shapes. The second part is that you need to now match the tRNA anticodon, which is attached, the amino acids attached now to the tRNA, with the mRNA codon. All right, so we have our mRNA, and this has a sequence of 5' to 3' ACG. So what's our tRNA anticodon going to be? Well, it's going to be the complement, and it's also going to be antiparallel. So here's the 5' end. Now this is just the loop of the tRNA, but up at the top of this whole structure, or the strand of tRNA, is going to be the 5' and the 3' being complement or antiparallel to the mRNA. The most important thing for us, though, is to recognize that the G is going to go with a C, the C will go with a G, and the A will go with the U, because this is RNA still, so U's are in RNA, not T's. So what's the anticodon? UGC is going to be the anticodon. That's 3' to 5', reading it, to the mRNA. And so if you saw something telling you that the anticodon was UGC, then you know it goes with an ACG mRNA or vice versa. There is a phenomenon we call Wobble, which means that some tRNAs can bind more than one codon. And it's always in this third spot here. So not all, but sometimes that third spot can match up with a non-complementary partner. So like for example here, AGA should always be UCU, right? Which it does bind to. That's the anticodon. But it turns out that AGG here, the G can also bind with UCU, and you wouldn't expect U and G to bind, but it's that third spot, and just the way that the shapes work, it can be flexible. And that's useful because it means one UCU tRNA can bind with and bring in arginine. So all the UCU tRNAs have arginine on them. And so that's one of the things about this redundant genetic code, is that AGA brings in arginine and AGG brings in arginine. And that's because of the Wobble. In order to facilitate the tRNA binding the mRNA, that's what the ribosome does. So let's look at ribosomes. They're similar in bacteria and eukaryotes, but they do have some differences, like their size. And the differences allow us to use antibiotics that can target prokaryotic ribosomes, bacterial ribosomes, to prevent them from working, but they don't hurt your own body because they don't hurt your eukaryotic ribosomes. Okay, so here's a protein type structure. One subunit, it's the large one in blue here, and then the small subunit in yellow here on the bottom. And we have three main spots. We have, and I'll start from the right side because that's what the order they're actually used. We have the A spot, we have the P spot, and these are pockets. We have the E spot. And I'll just tell you how I remember them. They have official names, but this is how I do it. The A spot, or the A site, is the amino acid site. That's where the new amino acid comes in. You can see that happening here. This is the new amino acid. The P site is the polypeptide site. That's where the polypeptide is growing out of, okay? And the E site is the exit site. That's where the empty tRNA is going to go leave and get reloaded with the new amino acid. The subunits don't exist together all the time. They're separate until the small binds the mRNA, and then the large comes on. And then you can see this polypeptide is growing out this exit tunnel, so you can see that here. And that's how it gets out of the ribosome. And eventually the whole thing will fall apart and be released. Okay. So here is this very simple version of initiation. You have, like I mentioned, the small subunit of the RNA binds the mRNA. It always binds at the 5' end, and then scoots along until it finds what we call a start codon. So this is mRNA on the bottom here. The mRNA start codon, which tells the ribosome where to begin, is AUG. So that's the sequence, AUG, AUG. So wherever the first AUG is in the RNA, the mRNA, that's where the reading will start. So if you actually have a mutation and then it changes this to like AUU, the ribosome won't recognize this as a start site, and it actually won't start until it reaches the first AUG. So that would actually make the protein missing, or the polypeptide missing the first part of it. So that kind of mutation would be a big deal, to put a different start codon in there or lose the start codon. You can see it's complementary to the anticodon here, and the anticodon always brings the methionine. Now, does this mean every polypeptide starts with methionine? Initially, yes. Then during the processing in the ER and the Golgi, sometimes that methionine can be cut off. But initially, all polypeptides start with this methionine from the AUG. Then you have your large subunit come on, and now you have an assembled, and the first tRNA is in the P site because it's ready to build the polypeptide, and that's your first initiation complex. So now we're ready to go. I should mention this word, reading frame. So that's, what are the groups of 3 that you're reading? Okay, here's the idea of the reading frame. This is your 5' end of the mRNA, and this is the 3', not the end, this is way too short, but this will be the 3' end eventually. So the ribosome is going to jump on the 5', it's going to scan until it gets to the first AUG. In this case, that's here. Then it's going to read groups of 3, because codons are 3s. So it's AUG, UAA, UGA, CCC, etc., until it gets to the stop codon, which we'll see what that means later. What if the sequence was like this? So all I've done is, instead of a G, I have a U here. So if the ribosome jumps on the end, it's going to read until it first gets to the first AUG. Where's the first AUG? Now here. So what's the reading frame? AUG, ACC, C, whatever. So you can see they'll be in the groups of 3 still. But look at this. Now instead of AUG, UAA, UGA, CCC, now we have different amino acids, still a methionine, but now we have an ACC. And you see it's this one here. But now it's shifted because we started at a different AUG. So that reading frame is now different. And so it's really important where that first codon is to start that reading frame in a specific groups of 3. This is elongation. That's when the polypeptide's built. All right, so what's going to happen? We have the new amino acid go into the A site. Then, interestingly, the polypeptide gets transferred onto the new amino acid. So it's going to bind onto the new amino acid. You can see now the whole polypeptide chain is on what's that new tRNA amino acid. Now this old one in the P site is empty, so it's going to come over to the E site, and the A is going to now be in the P site because that's the polypeptide chain is now in the P site. The E is going to exit because it's empty, the tRNA. Then you'll have an open A site, which the next amino acid comes in. And recall, why doesn't just any tRNA end up in the A site? Well, they can come in, but they won't stay unless the anticodon is complementary to the codon that's in the A site. So that's how it ends up getting matched up, and like I said, that brings the specific amino acid. Then we have termination. So there are 3 sequences that are stop codons: UAG, UAA, and UGA. You don't need to memorize those, but on the wheel, when you look at a codon wheel or a codon chart, it'll say stop, because those do not code for a tRNA. They code for matching. They match the codon with this release factor. And so what happens here is that if you try to transfer the polypeptide chain onto it, it doesn't transfer because there is no amino acid there. And so that releases the polypeptide chain. The empty tRNA exits like usual, and then the whole thing falls apart. That's not the end of the process, though, because more ribosomes have jumped on the mRNA at that 5' side as the other ribosomes are moving along or pulling the mRNA through. So you end up with a polyribosome, which just means, or a polysome, which means many ribosomes reading that RNA, and you can see each of them is making a polypeptide. You can tell which order they've gotten on because the polypeptide is the shortest on the newest one, and then the longest on the oldest one. Or you can see how much longer it is here. And also just note that the amino acid on the order is the same for all of them. Of course, not all of them have been added. And where are they adding? Remember, they add onto the site here. in the middle of the ribosome. That's where the action is at, in the ribosome. And actually, it's the rRNA, ribosomal RNA, that's doing the actual reaction. So if we looked at this amino acid out here, it's actually all the same amino acid. And what about the second one? It's the same for all of the groups because they're all reading the same mRNA. What about the third one? It's the same, and so forth. So that's why the sequence, of course, is going to be the same for all of them in the end. And so in both eukaryotes and prokaryotes, we have the polysomes. And in prokaryotes, we actually, or at least bacteria, we even have this additional dual step. So you can see the polysome here, right, or the polyribosome, where you have the mRNA in red, and then you have the ribosomes reading it to make the polypeptides in purple. And what's going on here, though, is that even at the same time, you have the transcription happening. This is the RNA polymerase. This is your DNA in blue. So it's all simultaneous, and you can see this complex down here on the right side started a lot earlier than the one on the left side. You can see there's lots more ribosomes, and the RNA, the mRNA in red is much longer because it's been reading the DNA for a longer period of time. So the gene is actually, the DNA, the gene is being read, transcribed into the mRNA in red. The red mRNA is being translated into polypeptides in purple, all simultaneously. Now this can't happen in eukaryotes, though, because the nucleus separates where the ribosomes are and where the DNA is. And then how do you tell where the ribosome started? Well, look how short this polypeptide is and how long this polypeptide is. So which one has been growing longer for a longer period of time, the very long polypeptide, which means that the ribosomes start here. And that means that's your 5' end. This also makes sense because what's going on here, that's where the action is at, building the mRNA, right? This is where the RNA polymerase enzyme is. So where, we know that mRNA and DNA can only be built on the 3' side. So we know that this has to be the 3' side of the mRNA because that's where the action is at. And this makes sense because we know ribosomes jump on the 5' side. So that all works out okay. And you can see a real picture up here. You can target the ribosomes to be releasing their polypeptides into the reservoir. So we talked about bound ribosomes. So this is a bound ribosome. And why does it end up being a bound ribosome instead of a free ribosome in the cytosol? Because the RNA has part of it that codes for 3 amino acids, or I should say a few, codes for a few amino acids at the tip. And those amino acids are what we call a signal peptide, or signal part of that polypeptide. And they basically bind other proteins and target that polypeptide to the reservoir. So there's these receptors on the reservoir, and if that little peptide part binds it, then the protein will be either dumped into the reservoir, or polypeptide will be dumped into there and get folded into a protein, or it might not be threaded all the way through, and that's how you end up getting those proteins that would stick in the membrane. Remember, we saw that that could be a final destination of a protein, is to end up being in a membrane of like the cell membrane. So this would be the way that happens. Now this ribosome is not always bound, because when it falls apart, now it's like, ribosome parts again, and depending on what mRNA it binds, dictates what kind of sequence it builds and whether it's targeted or not. We can regulate translation by degrading the mRNA, so actually breaking it down. And microRNAs or small interfering RNAs can target mRNAs for actual breakdown, or we can physically, some types of miRNAs, microRNAs or siRNAs actually block the ribosome from getting on. So those are 2 ways that can happen. You can see mRNA degradation here. So some types of mRNAs are going to do that, induce the chopping up of the mRNA, and sometimes they'd actually block the ribosome from binding. But in both cases, interestingly, that means your gene is getting expressed as far as transcription goes, but you never get a polypeptide. So really the gene isn't expressed overall. So this is how we'd stop gene expression at the translation level, is that you either get rid of the mRNA or you stop the ribosome from working. These are both what we call non-coding RNAs. There are lots of types of non-coding RNAs. Most of the genome is not coding for proteins. A lot of the genome consists of these non-coding RNAs, which are like microRNAs and small interfering RNAs, piRNAs, link RNAs. Some of them are specific to stem cells, some of them are just general regulation. So the microRNAs and small interfering RNAs are ones that are regulation. So they stop gene expression. We can utilize this in a process called Rnai, which basically you take siRNA and you inject it into cells, or you inject it, or you put it in the food of like these tiny nematode worms, or you can put it in the food for other organisms, or put it into the developing embryos, and it blocks gene expression. And you specifically design the siRNA you want to use, and that would basically turn off the gene, and you wouldn't even have to manipulate the genome. They also have used some RNAi for human disease. So if the problem in the human disease is a gene being expressed, so it's expressing some sort of protein that's a mess, let's say, and you want to stop that expression, then there have been some progress in using this. You make your own siRNAs, which is like an miRNA, and you make your own design to target the RNA, the mRNA that is a problem, making the messed up protein. And so if you basically stop the mRNA from being translated, then you stop the expression of that messed up protein. And this has been successful in some sorts of diseases. And then just to mention mutations, there are substitution mutations. So you can see here the original sequence had a G at that spot, and now we have an A at this spot, which now in the DNA, once it gets copied, that's going to be with a T. So it's an AT pair instead of a C or GC pair. So what does that do? Instead of putting in a C, it's going to put in a U during transcription. And then in translation, what happens in this case, GGC and GGU both code for glycine. So this is a silent mutation where there is no actual effect on the final protein product. There's no effect on that polypeptide because of that wobble idea and the fact that you have redundancy. Here's another substitution where you have a TA pair instead of a CG pair. Let's see where here's a CG pair. So in this case, AGC codes for serine instead of GGC coding for glycine. So this would be called a missense mutation, where you have a mistaken amino acid, one or more. So a mistake amino acid or a different amino acid, one or more. And this is not necessarily bad or good. In fact, it might not even really affect the organism. But if you have that, it's a missense. Here's another substitution where we have an AT pair because there's a mutation here, and instead of the A, a T was put in. So it's the opposite, right? And that matters because usually there's an A at that spot. Now there's a U because only one of the DNA strands is used for transcription. And what happens here? UAG. So we have an early stop codon. When you have an early stop, it's called a non-sense mutation. Usually it makes a short, well, it always makes a short polypeptide. Often that just gets degraded and doesn't fold. So basically you have no protein product at that point. Those are substitutions where you're switching out a nucleotide by accident. Then there are also indels, insertions, deletions. So here's an extra A. You can see, let me erase this. Let me just highlight it. You can see before, here's the TTC, but now there's an extra A that was put in during DNA replication by accident. And that means that there's now a U that's in there, and now we have a UAA, which is a stop codon. So this would be insertions and deletions always cause frame shifts. So frame shift means that the reading frame is now different, right? So you could see that the reading frame shifted over because now our reading frame is UAA instead of AGA. You can see the AAG. So here's the AAG. That's what used to be red, but we shifted the reading frame because we had an extra U. Same thing with the deletion. You'd have a shifted reading frame. So there's not literally a hole in the mRNA. and that's the cell doesn't know any difference than what it usually is. But here you see UUG. Previously it was UUU, but accidentally during DNA replication it jumped over the A when I was replicating, so now that's no longer there, which means the U doesn't get put in. Which means in this case we have different amino acids, and in fact all the amino acids after the insertion or deletion, in this case deletion, are going to be messed up. So if you have an insertion or deletion of 1 or 2, everything after it's going to either be missense and all different amino acids, or if you have a stop codon put in, then it would be a nonsense mutation again. And a special case is if you have 3 deleted or 3 that are added during DNA replication and they're in frame, like they're just added between 2 codons, you could just get a new amino acid or missing amino acid. In this case, it's deleted. So you see everything's okay except you're just missing lysine because that TTC is no longer there. So in that case, you might not have too much of a problem. It just depends. All of these just depend. I'd say frame shift causing extensive missense is probably a disaster. Now, does that mean a disaster as in the organism's in trouble? No, because sometimes losing gene expression or losing a protein product is advantageous. So it's not that, having a mutation that causes a major protein problem will be bad. It's just that that protein probably won't work anymore with extensive missense. Also with nonsense, if it's short, the protein's probably not going to work anymore. Whether that's good or bad just depends on the scenario the organism finds itself, as well as what it does to the physiology, anatomy, biochemistry, something like that. So that's what I want to tell you about translation.